Beepsa, we can't hear you.
Hi, Ilya. Yes, I'm going to be starting in 1 more minute. Thank you.
That's true.
Hello, everyone, and welcome to this special session on research project proposal by our research fellows and Omics Logic program participants. The goal of the Omics Logic programs is to provide training in bioinformatics, enabling independent research guided by mentors and peers, and enabling students and researchers to develop novel and independent research using data science. To achieve this, Omics Logic programs follow a project-based learning approach using research-grade tools to analyze data from top peer-reviewed journals. This Omics Logic training has been completed by over 10,000 participants from 162 countries and over 300 workshops for 6 specialization tracks like oncology, infectious diseases, precision medicine, neuroscience, data science for biomedical data, and comprehensive training on omics data analysis. As a result, anyone starting on this journey in bioinformatics can start developing a project of their own. In fact, we have already worked with many students who have completed this kind of training and developed interesting research projects that won awards and got published in peer-reviewed journals. Many of our past participants also had a chance to present their analysis in front of their classmates or at conference poster sessions in workshops. As we start with a review of the presentations today, I want to introduce you all to Dr. Harpreet Kaur, who would be sharing her feedback on the projects. Dr. Harpreet Kaur is one of the trainers who helps to facilitate many of the sessions. Her area of specialization is in genomics and machine learning. She has over 16 peer-reviewed publications and has been monitoring and mentoring the Omics Logic program participants over the past several months. Now, let me introduce you to some of the projects by our research fellows focused on Alzheimer's disease. The Research Fellowship Program has been designed to help young researchers and students take advantage of the bioinformatics resources for analysis of complex, high-throughput life science data and become versed in bioinformatics. So this program offers you to participate in real and cutting-edge bioinformatics research and be part of a renowned bioinformatics community. The first research fellow who is going to be presenting is Andres. So let us see what Andres has to say.
Okay. My name is Andres Moreno Sosa. I'm a high school student from Mexico City, and I'm currently doing the
Beepsa, it seems like we cannot hear. You muted yourself?
Yeah, okay. I'll unmute myself.
Genetic diseases, namely Alzheimer's disease. And my project is titled, at least for the moment, Evaluating the Role of Microbial Cells in the Pathogenesis and Progression of Alzheimer's Disease. And my mentor is Dr. Harpreet Kaur. So Alzheimer's disease is an irreversible and progressive brain disorder that results in the development of cognitive deficits and clinical dementia. It is characterized by the development of 2 pathological hallmarks, the first being the development of extracellular amyloid beta plaques, and the other one being intracellular neurofibrillary tangles. Although these are widely regarded as the 2 main pathological hallmarks of Alzheimer's disease, scientists still don't truly know how they link to each other. And because of these, They believe that there may be other pathological mechanisms that underlie, the pathogenesis and progression of Alzheimer's disease. It is widely known that patients with Alzheimer's disease show an increased inflammatory response after they start developing amyloid beta plaques. And this inflammatory response has also been shown to exacerbate the development of both neurofibrillary tangles or tau pathologies, as well as the development of these amyloid beta plaques. And for this reason, some scientists believe that inflammation might be the third core pathology of Alzheimer's disease, thus providing a link between the other two main pathological hallmarks. Inflammation in the brain is mediated by microglial cells, which are the brain's resident macrophages, which are part of our body's immune system. And because of this, they have been shown to mediate neuroinflammation in the brain by the secretion of pro-inflammatory cytokines. And yes, I had said, so scientists believe that this neuroinflammation mediated by microglial cells might constitute this link between the development of amyloid beta plaques and the further development of neurofibrillary tangles. So for this project, I wanted to evaluate how the transcriptome of microglial cells varied across the age, Plasmodium age. And I used this data set from this study, transcriptomic profiling of microglia and astrocytes throughout aging from the Journal of Neuroinflammation. So in here they used wild type and APPPS1 mice. APPPS1 mice show an elevated development of amyloid beta plaques because they overproduce the amyloid beta peptide. So they show a higher development of these plaques in here. And they used 58 samples. So they sequenced the transcriptome of both microglia and astrocytes in both wild type and AD mice. The AD mice are the Alzheimer's disease model mice, so the APPPS1 mice. And they sequenced the transcriptome at 5 different time points. So 2 months, 4 months, 6 months, 9 months, and 12 months. And in the end, they had 58 samples. I believe that the data from this data set provides valuable information regarding microglial activation in response to aging and amyloid beta pathologies, because as I had said, these mice, APPPS1 mice, they show elevated levels of amyloid beta plaques. And so we can really see how these amyloid beta plaques are changing the transcriptomic profile of microglial cells, and to see whether there is truly a neuroinflammatory response due to the development of these pathologies. So based on these files they provided, I decided to just focus on the microglial cells. I performed an RNA-seq pipeline. So I quantified the gene expression levels of microglial cells, both across the wild type and the AD mice. And just as with any RNA-seq pipeline, I pre-processed the data, then I mapped the RNA transcripts to the organism's reference genome, and I then quantified the abundance of reads aligned to the reference genome so that I could get a gene expression table in read counts, in raw read counts. And well, yes, I obtained the expression levels in raw read counts as an output. And then I used this gene expression table as an input for several differential expression analyses. And well, yes, based on this gene expression table that I got, I performed the following differential expression analysis using this two. So the first one was young AD versus old AD. So this will allow me to see how the transcriptome of microglial cells vary in pathological conditions as the mice age. Then young wild type versus old wild type. This constitutes a reference to which I could compare the first differential expression analysis as to see whether mice that are not in pathological conditions exhibit the same changes across staging, or if the changes that I observed in the first differential expression analysis are just inherent to the disease, or yes. Then I did young wild type versus young AD to see whether the transcriptomic differences between microglial cells in pathological and physiological conditions arise like from a young age, or whether they arise as the mice begin to age when they are older. And then these last one, old wild type versus old AD, to see the transcriptomic differences between old wild type mice and old AD mice, and to see like what are the main physiological differences between these two clinical groups. And just to like explain, I defined young mice as those that were 2, 4, and 6 months old, because based on what I've read, these mice start developing amyloid beta plaques around 6 to 7 months old, when they are 6 to 7 months old. And so I thought that it would be, based on this observation, I should include the young mice as 2, 4, and 6 month old mice, so that the old mice would be those that showed a truly elevated level of amyloid beta plaques. And then after performing the differential expression analysis, I filtered the differentially expressed. I selected the differentially expressed genes for each analysis, and you can really like. I think that this provides us an insight into when the transcriptomic changes, if you could call them that, arise. So you can see that between the young wild type and the young AD, we only have 9 differentially expressed genes. So that really tells us that they are rather similar to each other, and that the physiological differences appear as the mice begin to age. And then when we compare the young AD to the old AD, we can see that there is a greater number of differentially expressed genes in comparison to the young wild type versus old wild type. Of course, we also see a rather elevated number, but this one's larger, so that maybe tells us that there is a greater physiological divergence in terms of the transcriptome. Yeah, and so based on these differentially expressed genes that I have gotten, I performed 4 different principal component analysis with those selected genes, so with the differentially expressed genes. So this one in here is from the young AD versus old AD. So here we can say that the old AD mice are clustering to the left side of the plot, whereas the young mice, so those from 2, 4, and 6 month old mice, are clustering in here. So these are all AD mice. So here we found that one of the 6 month old mice is clustering like towards the old mice, which honestly isn't like that surprising, because as I had said, these mice start developing the amyloid beta pathologies around like 6 to 7 months old. And so maybe these mice starting developing these pathologies when it was perhaps like younger, because of course it varies. It isn't like that when they are 6 months old it starts. But yeah, then here I performed the young wild type versus the old wild type. Again, here you can see that the 2, 4, and 6 month old cluster on here, whereas 9 to 12 month old cluster on here. But we don't see like such like it isn't that polarized. And then from the differentially expressed genes, I selected those that showed, using log2fold, those that were like overexpressed and those that were underexpressed based on this differential expression analysis. And I could see that in the young AD versus the old AD, and also in the old wild type versus the old AD, we can see an increase in the activity of neutrophil degranulation, neutrophil activation involving immune response, neutrophil mediated immunity. And here we can see those same things. And then we can also see here the inflammatory response, positive regulation of acute inflammatory response, positive regulation of inflammatory response, regulation of inflammatory response, positive regulation of cytokine production. So these are all mechanisms involved in immune processes and inflammatory mechanisms, which reinforces what I had stated in my introduction. And when we compare that, when we compare these differences to the differences that arise when comparing the young wild type versus the old wild type, we see that in here there will be an inflammatory response, or like an increase in the immune processes. So that really tells us that this is inherent to the pathological conditions, because we are not seeing that in here. And yeah, as I had said, the main difference between the wild type mice and the APPS1 mice is that the latter overproduced amyloid beta, and so they showed increased levels of these amyloid beta plaques. So this could tell us that the aggregation of these amyloid beta plaques likely constitutes a cause of microglial immune activation. And as I had previously said, this immune activation, this neuroinflammatory response in the brain, has been shown to exacerbate the development of neurofibrillary tangles. So this really reinforces this link between amyloid beta plaques and neurofibrillary tangles, and that link is like this neuroinflammatory response. And yeah, so based on these mechanisms that were the most significant, so neurotrophil degranulation, neurotrophil activation, neurotrophil mediated immunity, I tried to research a little bit about what are neutrophils. I'm sorry, I think that I had said neurotrophils. It's neutrophil. Neutrophils are the most abundant leukocytes, so they are white blood cells in the circulation. They modulate immune processes, and they've been shown to play important roles in pathological inflammation. And they are granulocytes, which means that they are a type of white blood cells that contain specific granules, which are some like vesicles that contain cytotoxic molecules as well as pro-inflammatory cytokines. And then when they became activated, they can produce reactive oxygen species, which also promote inflammation, and it can also lead to phagocytosis and the secretion of pro-inflammatory cytokines. Yes. And so what's next? So as I had said, amyloid beta aggregation may induce the expression of and the secretion of pro-inflammatory cytokines via microglial cells in Alzheimer's disease. But now I want to investigate like what are the cellular and molecular mechanisms that regulate the expression of these cytokines. Long non-coding RNAs are key gene expression regulators in both innate and adaptive immunity processes. The function of these molecules in pathological processes, however, remains largely unknown. And they have been shown to regulate the inducible expression of cytokines after immune activation. So now I want to answer this question. What are the roles of long non-coding RNAs in the pathogenesis and disease progression of Alzheimer's disease? Are they involved in the regulation of microglial activation states and neuroinflammation in Alzheimer's disease? So that is all. Thank you for your attention.
So thank you everyone for listening in to Andres's presentation. If you have any questions for Andres, then please let us know. And Ilya and Dr. Harpreet, if you had anything to add, so please add to Andres's presentation.
I think it is a nice presentation, and he's already completed a lot of work, so I think it is very nice presentation. And maybe some of the text he can decrease on his presentation.
Okay, thank you so much, Dr. Harpreet, for that feedback. If you don't have any other questions from the participants attending this session, I will move on to the next slide. So the next presentation I have is again by a participant who is not present today. So this presentation is by Netra. So both Andres and Netra is not present because they have school today. So we are going to be seeing what they are coming up with. So I'll play Netra's presentation now.
Hi, everyone. My name is Netra Srinivasan, and my project is the effects of the onset of menopause on the transcriptomic profile of the aging female brain. First of all, to start off with the significance of the problem, there appears to be a divergence in the occurrence of neurodegenerative diseases between men and women, but the mechanisms so far are unknown. For example, for Alzheimer's disease, more than 5.5 million Americans are living with Alzheimer's disease, of whom 2/3 are elderly women. And it's interesting because there is protection against neurodegenerative disorders for younger women. Like, for example, mitochondria from younger females are protected against beta amyloid toxicity, generate less reactive oxygen species, and release less apoptogenic signals than those from males. However, the advantage is lost in the mitochondria in older females, which happens with the onset of menopause. And furthermore, current Alzheimer's treatments only target acetylcholine and glutamate to treat symptoms, but not the root cause. And since there seems to be a divergence and the discrepancy between the two genders, it's important to figure out the underlying causes to be able to better target treatment. So first of all, I'd like to figure out what is going on as the female ages and if there are any transcriptomic differences that can help us identify genes at play. And this, which can also help us pinpoint pathways that related genes may play a role in order to target treatment effectively. Next, to talk about female aging for background information. So menopause is the end of a female's menstrual cycle and fertility, in which the ovaries no longer make estrogen and progesterone. And this does not happen just in humans, it happens in most mammals as well. So in this project, the data is collected from rats. And so we will be looking at the transitioning of menopause in rats. So the first phase, which is both in humans and in rats, is premenopause, which is about 6 months for the rat, in which they have normal menstrual cycles. And they transition onto perimenopause at about 9 months, in which the cycles become irregular, but they haven't stopped completely. So menopause at about 9 months is starts with the final menstrual cycle. And for humans, it would have stopped for about a year when we think about human menopause. And then post-menopause is at about 15 months for rats. But this is in humans when we hit the first year mark from our final menstrual cycle. From previous studies, as the brain ages in women, there's also an increased immune response. And so we are obtaining data from this study by Mishra et al., in which we have 6 groups, 6 samples each. And this is using RNA sequencing from the Rattus norvegicus, female hippocampus. And so we have 6 groups in which there's the female rat at 6 months, and it's abbreviated as Reg6M because they have a regular cycle of 6 months. Then there's 9 months with the normal menstrual cycle, or Reg9M, 9 months irregular or irregular 9M, 9 months acyclic or acyclic 9M. Then OVX at 9M, which is overrectomized rats, which means that their ovaries have been removed. And this is significant because, and it acts as a control, because OVX represents the most important clinical features of estrogen deficiency induced modeling postmenopausal conditions. And then there's also the female rat at 16 months acyclic menstrual cycle. And so as we continue on, I found my research question and hypothesis. So firstly, understanding the genes expressed differently in the progressive stages of female aging and menopause can help us understand risks for neurodegeneration and pathways to target in order to treat neurodegeneration, such as for Alzheimer's disease in women. So a couple of questions that I had were, how does the age or the time of the onset of menopause affect the transcriptomic expression, and which pathways are expressed? Are there biomarkers that help us identify the risk for neurodegenerative disorders? How does the expression differ in overrectomized species and regular specimens, and how can that lead to insights in the onset of neurodegenerative disease? And overall, my central question is, what are the effects of the onset of menopause on the transcriptomic profile of the aging female brain? So my hypothesis is twofold. Firstly, I hypothesize that as the females enter premenopause and perimenopause, they will express less genes related to neurodegeneration as the females in menopause and postmenopause. And secondly, I hypothesize that the age and the age of the onset also have an effect on the transcriptomics of the rat. So basically, the earlier the onset of menopause, the more likely they're going to be expressing the transcriptome for neurodegenerative disorders such as Alzheimer's disease, which we can compare with those different groups. So for my methodology, I had 6 groups to compare, and I will be using principal component analysis, differential gene expression analysis, and hierarchical clustering. And I will be using those to relate expressions to each other to draw conclusions, analyze which genes are underexpressed and which are overexpressed in 6 groups, and look at significantly expressed genes and annotate their function on David and Enricher to see the pathways expressed, such as with neuroinflammation or neurodegeneration, etc. And for my results, first of all, I did preliminary PCA between the 6 groups, and I found that there was a divergence between the regular groups and then the groups that were either irregular or acyclic. And so therefore, we then grouped the regular groups and the acyclic groups and the irregular groups. And then I did PCA. And these are the PCA graphs with all of the genes. And so between regular versus acyclic, we can see that there are 2 groups here. And for regular versus irregular, it's not that they're not 2 specific groups because the components are very small. And they aren't filtered yet with only the significant genes. But I went ahead and I did differential analysis and with the further PCA. So I compared the regular versus the overoptimized, and I found that they were significantly different. And so, and I also used the significant genes to make these 2 PCA plots in which we can see that there are 2 discrete groups showing that there is a very pronounced difference between the regular and the irregular and the regular versus acyclic. And that even despite the same age, there is a difference in the transcriptome when it comes to what the regular versus the postmenopausal conditions, meaning that menopause has an effect on the transcriptomic profile. So with differential expression, with further, I did the volcano plots and I found these significant genes. And also I've got more plots for the regular versus acyclic and the regular versus irregular. And to summarize for what I got with the significant genes, the pathways that were upregulated were the regular versus irregular and regular versus acyclic were related to immune function. And for downregulation, it was related to oxidative phosphorylation, which is linked to neurodegeneration, and related to mitochondrial dysfunction leading to neurodegeneration. And for regular versus irregular and regular versus acyclic, they actually share for downregulation linked to higher neurodegeneration, and also a higher immune response. And for the hierarchical clustering, when I did the hierarchical clustering, I also my theories were confirmed that that the 2 groups were significant. And so through the pathway analysis on Enricher, here is the visualization of the expression of the pathways, in which over here we could see it relate to immune function, and over here is for like increased neurodegeneration related to mitochondrial dysfunction and oxidative stress. Same thing over here for regular versus acyclic, a heightened immune response and a higher neurodegeneration linked to mitochondrial stress. So to conclude for my results, I found that there was indeed an impact in the transcriptomics of the rat, especially in relation to the mitochondrial function leading to the neurodegenerative diseases such as Alzheimer's and Parkinson's disease. And I also found that there was a heightened immune response. And so this leads to the conclusion that because of the onset of menopause, this leads to the targeting of possible treatments for neurodegenerative diseases caused by oxidative stress. So this is confirmed that from this data that menopause, that the onset of menopause somehow has an effect on the oxidative stress, causing different neurodegenerative diseases like Alzheimer's disease. And it also supports the current pathologies of Alzheimer's because for Alzheimer's, the oxidative stress that is exacerbated damages the mitochondria, which leads to cell death and even more accumulation of beta amyloid and tau proteins and neurofibrillary tangles. And in addition, it's something interesting that the oxidative stress can actually be reversed. So when there's oxidative stress, the mitochondria structure is damaged. However, researchers found that when the causes of the oxidative stress is removed, the mitochondria structure bounces back, which could lead to a possible treatment. Now, for the limitations, certain limitations is that I was not able to extract this information myself in the lab, and I had to use data that other researchers extracted from their samples. So I was not able to control the sample size or the procedures used to collect the data. Therefore, I'm dependent on their collection strategy to obtain my results. And for further research, I'd like, if I was given the opportunity to go into a lab, I would possibly see how estrogen and progesterone supplements would then affect the transcriptomics of the rat, since in menopause, the rats are estrogen and progesterone deficient. And so by testing that, that would confirm my results. And I would also like to see if this is true in humans as well. So thank you for listening to my presentation.
So thank you all for listening into Netra's presentation as well. So Dr. Kaur, do you have anything to add or a feedback for Netra? Thank you.
I think a good presentation. There are some points there which need improvement. I think I can email her regarding that, and that is regarding one of the PCA plot. And rest of presentation is, I think, quite good. She's already completed a lot of things. So some of the issues that she can address, we can actually ask her. So I think rest of presentation is good.
And here is a question from Emmanuel. Spliceosome-related genes or pathways were overexpressed. Any implication?
I mean, I would just like to add, maybe Harpreet, you can answer that in just a second. I just wanted to add to let everybody know that these two projects were developed by high school students who are developing their research projects to participate in life science fairs and competitions. So I think that's something to keep in mind as we review these.
Yeah. Thank you so much, Ilya. Dr. Harpreet, yes, you can go ahead.
I think spliceosome, maybe there are some of the isoforms that are generated, eventually some of isoforms that affect menstrual cycle. Maybe the spliceosome related genes play an important role in that case.
Thank you, Dr. Harpreet. So for the next presentation, we have our intern Sonalika Ray, who would be presenting on the analysis of liver cancer transcriptomic data for drug repurposing. So Sonalika, please go ahead. Thank you.
Thank you, Deepsha. Good evening, everybody. I'm Sonalika Ray. Currently, I'm pursuing Masters in Bioinformatics in Punjab University, and I'm interning with Pine Biotech. And my purpose here is to complete my dissertation project for Masters. My research interest lies in genomic data science and computer-aided drug designing. So today I would be giving an overview of the literature and would discuss the highlights of my research proposal, which is on the analysis of transcriptomic data for drug repurposing. So you see, HCC is the most common form of liver cancer, and it is the third most leading cause of death worldwide. It has got various risk factors involved with it. Also, there are about 7 to 8 drugs which are actually in the market to treat HCC, but it's only the drug sorafenib, which is FDA approved in US. So sorafenib is such a drug which actually doesn't show its effect in the later stages of HCC. And therefore, the HCC becomes sorafenib resistance. So HCC remains a treatment challenge due to, first of all, late detection. Yes, because most of the times HCC is detected very later in the stages and resistance to currently approved drugs. So the paper which I went through was the transcriptomics-based drug repurposing approach, which identifies novel drugs against sorafenib resistance hepatocellular carcinoma. And the purpose of this paper was to expedite identification of novel drug candidates against sorafenib resistant SCC. So the workflow as shown here gives an overview of computational drug repurposing and drug target network. Herein, the SR-SCC, that is the sorafenib-resistant SCC cell lines, were generated from the normal SCC cell lines by exposing the normal cell lines to sorafenib for a period of time And then the gene expression signatures of experimental models of SCC sorafenib resistance were assessed. They were assessed for prognostic significance, and the queries against gene expression signatures characterizing drug perturbation in HepG2 cell line containing the LINCS database. Now, the LINCS is a database that is Library of Integrated Network-Based Cellular Signature Analysis. This LINCS database provides us with the connectivity scores, which were calculated by the rank-based non-parametric weight Kolmogorov-Smirnov statistic, and drugs with negative connectivity scores, that is anti-correlated, represent that are hypothesized to reverse SCC sorafenib resistance gene signature. And the drug candidates were further prioritized based on FDA approval, clinical investigation status, known as antineoplastic activity, and literature evidence for drug target genes associated with SCC. And then finally, 2 drugs were subsequently selected for in vitro validation. So this is just a very small representation of what the workflow actually was. So here 2 cell lines were taken. This first one was a normal SCC cell line, and then we had the sorafenib resistant SCC cell line. And then these 2 cell lines were subjected to connectivity mapping through LINCS database. Now, connectivity mapping provides with the connectivity scores, which were calculated by the rank-based non-parametric weight KS statistics. And after the connectivity score, the connectivity mapping, the data sets were taken from TCG and GEO to cross-verify with the gene expression data sets characterizing SCC patient tumors of diverse etiologies. And this is just a look of what the LINC tools look like. So there are various tools which are involved with the LINCs, and I would be reviewing them later in the presentation itself. So my research question lies to search for molecules, which could actually be used effectively against the sorafenib resistance SCC. And then I would like to find the pathways which are involved with these molecules, those pathways which these compounds actually affect the drug targets. So I have few questions which I would like to answer while linking the effective compounds against SR resistance SCC. That would be, out of all the compounds, which compound could be linked to SCC, and whether the predictive compound was effective. And if it was effective, then in which pathway it was involved. And the approach I would be following would be to take the cell line and then match the gene expression alterations in resistant SCC with gene expression changes, which are caused by the treatment of cancer cells, with drugs already FDA approved for other diseases, and then find the drug that can reverse the resistance-related changes. Herein, approved and investigational drug link files could be downloaded from drug bank database. And to look for drugs under clinical investigation, we can go through AACTB database, that is Aggregate Analysis of Clinical Trial.gov database. And then the KEGG drug database IDs could be mapped to drugs in drug bank to extract drug activity and target pathway. And then the drug target genes obtained could be assessed with SCC using the Beagle literature mining tool. So once we have the drugs which could be used effectively against the sorafenib resistant SCC, then we could look, or we could identify the mechanism of action, that is the pathway or the drug target which is involved in the SCC resistant sorafenib, right? And after we are done with the mechanism, then we could provide proof of concept evidence for validity of the drug repurposing approach with potential for use in the personalized medicine. So now I would just like to explore the links with you all and show you how it actually works. So here, this is the links tools database where a lot of tools are mentioned. So it has a long list of tools, but here I would be interested in the tools like repurposing app, which could be used for drug repurposing, drug cell line browser. So now this is a really very nice browser which could be used to actually look for various drugs and cell lines, and then a drug pathway browser, iLinks. So let's just have a look at iLinks. So you see this I-links, it helps us to give a list of gene signatures. So if I go for the HG2 cell line, then I would actually get a list of gene signatures, which I could easily download and look for the genes. Now, in this gene signatures list, I would be getting scores and probabilities, according to which I could select which genes I have to further analyze, right? And then I have another set. Just give me a second, sorry. Yes, actually, I'm not able to. Okay, so maybe that was it. Now I would like to hand over to Beepsha, ma'am. Thank you.
Thank you, Sonalika. Dr. Harpreet?
Well, just before Dr. Harpreet, I want to mention that, this project is very interesting and I think relevant for anyone who has an interest in drug repurposing, because what essentially she is doing is combining a transcriptomic data set with FDA-approved drug signatures on this LINCS database, and might be an interesting approach for some of the other people that I know are a part of this session who are thinking about different types of drug repurposing projects of their own.
So can I add something else as well? Actually, there was some issue with my screen. Now it's fine. So I actually wanted to share the screen and show like how wonderful this repurposing tool is. So if I look for the drug sorafenib and try to look at the target, so let's say I look for the BRAF target, and then I could easily take that target into drug bank and look for other drug relations which I could get. So if I look for BRAF itself, I get a drug named fostamatinib, which could actually be used as a potent drug, again effective for the sorafenib resistant cell line. So this way we could actually get a good list of drugs. And also then the other tool, which was a drug cell line browser, so I could easily download the sorafenib resistant SEC cell line of the liver from here, and with this download results button, I would actually get an Excel sheet of all the list of all the cell lines, and then I could use it further for research. Thank you.
Great. Thank you so much, Sonalika, for sharing this. Dr. Harpreet, did you have something to add to this?
Hello, Sonalika. I think this is nice presentation and good proposal. So I think you can proceed for this project. And there is some of the things that you can also find some other cell lines maybe that show resistance to the particular sorfanib like, right? So those are drugs against, you can actually find some other cell lines also in literature.
Sure, ma'am. I would. Thank you, ma'am.
Yeah, I think it looks feasible project.
And thank you, Sonalika, for presenting. In the next part, we would be listening to participants who have joined the Bioinformatics for Infectious Diseases Training and Research Program. Now, let me give you a little bit of a background to what this program was. In January 2021, we launched a 6-month research program in collaboration with the University of Ghana, focused on infectious diseases. This program is organized by Dr. Anita Gansha, who previously completed a research fellowship in several different Omics Logic programs. After completing an introductory overview of bioinformatics methods for infectious diseases research, we are now planning 2 follow-up programs on genomics and transcriptomics. So as our participants have completed the infectious disease program, many of them has research proposals which are ready. And today we are going to be hearing 3 such research proposals. So for the first part of this, I would want to invite Courage Adobor to present on genetic variation studies of sexual stage specific antigens PFS 230 and PFS 4845 of Plasmodium Falciparum population in Africa and their potential implications on transmission blocking vaccines. So Courage, please go ahead and share your screen.
Please, I cannot share my screen. Can it be enabled?
Yeah, you are co-host. Yeah, thank you.
Can the other user stop sharing so that I can share?
Hello, Courage. Can you go into presentation mode? Yeah, thank you.
So, hello, my name is Courage Adobo, and in this presentation, I will provide a rationale for the project titled Genetic Variation Studies of Sexual Stage Specific Antigen, PFS 230 and PFS 48 of Plasmodium Falciparum populations in Africa and their potential implications. Proposal has been developed as a part of the Omics Logic Bioinformatics training program conducted by Pine Biotech in collaboration with the Noguchi Memorial Institute for Medical Research at the University of Ghana. So I will be collaborating with Larry to do this project. My personal background is in bioinformatics. I have a BSc in biomedical engineering, and I have been interested in population genetics of Plasmodium Falciparum. Larry's background is in veterinary services, and he's interested in infectious diseases, animals breeding, and genomics. As the title said, this project will help understand the implication of genetic variation on transmission blocking vaccines against malaria. And this is being done under the supervision of Dr. Anita Gansa and the Pan Biotech team. Plasmodium falciparum malaria still remains the deadliest form of malaria, especially in children under 5. Several control interventions, including anti-malaria drugs and vector control measures like bed nets and insecticide sprays, have been used to prevent the disease. One tool lacking in this arsenal is a broadly effective malaria vaccine. Over the years, the malaria parasite life cycle has been tested and tried out, but they all face the problem of low efficacy, which studies have shown to be mainly due to high genetic variation of circulating parasites in the population. Notable among this vaccine is the currently most advanced malaria vaccine, which is the RTSS, which targets the liver stage of the parasite. So talking about the life cycle of the parasite, after the mosquito comes for a blood meal and then it inoculates sporozoites into the human host. So this sporozoite travels to the liver and infects the hepatocytes. And then the infected hepatocyte multiplies the parasites, and then further merozoites are released into the bloodstream, which goes to infect red blood cells. These infected red blood cells, merozoites, after some time, some of them mature into gametes, and these gametes are picked up by mosquitoes when they come for another blood meal. So the RTSS vaccine, which targets the liver stage, has also shown low efficacy of about 39% against clinical malaria, which was mainly due to the high genetic variation in circulating parasites, as shown by some studies. Some studies have also explored the potential of vaccine targets that can block the transmission of gametocytes from the human host to mosquitoes. which can further block the fertilization and halt reproduction of these gametes. Two antigens, PFS 230 and PFS 4845, which are expressed on the surface of gametocytes, have been shown to elicit strong antibody responses against Plasmodium falciparum. However, the problem of genetic variation, which plagues the other vaccine candidates, have not been fully explored within these two antigens in the African parasite population. So the problem, the efficacy of current malaria vaccine candidates are hindered due to the diverse nature of their target antigens. Successful transmission blocking vaccine will induce immune responses against the malaria parasite gametocyte sexual stages. However, the genetic variation of transmission blocking vaccine antigens have not been fully explored in the African parasite population. So there is the need to explore the genetic variation of transmission blocking antigen to further estimate the potential implication on transmission blocking malaria vaccines in the African parasite population. So our approach is to call single nucleotide polymorphism from raw data, analyze the functional effect of these SNPs, and further analyze the effect of these polymorphisms on epitopes, and then evaluate the population structure and differentiation within Africa. So we'll be taking our data from malaria gene. The approach in detail is split into three parts. So we have the processing, the analysis, and the structure and function. For processing, we will take raw read sequence data, run the data through quality checks, perform multiple sequence alignment with the reference sequence, call variants, that's specifically SNPs on the aligned sequences, SNPs in the genes of interest from the variant data, and further explore these genes, and then look at how they differentiate in the African populations. And then look at the functional effects of the SNPs when translated to amino acid. For structure and function, we will look at the effect of these SNPs on known and potential epitopes. And finally, do 3D structure analysis on the wild and mutant structures. Okay. So about the data set, we will take the community projects in malaria gene, specifically the raw reads sequences, which are saved in the European Nucleotide Archive database. This data contains 833 samples from 8 African countries and 17 sample sites. These samples were collected between 2014 and in 2015. Okay. So the graph shows the distribution of samples per country. And as you can see, some countries have a lot of samples. Thus, we will employ statistics to help cut down the samples to an acceptable number, which will not affect our analysis further on. And this snapshot tells us that data, this is a snapshot from the data table, which shows the metadata associated with each sample. Okay. Also, we will retrieve the 3D structures from protein data bank. With populations grouped into West Africa, Central Africa, and East Africa, this table shows us the overall derived allele frequency of SNPs within PFS 230 and PFS 4845. In general, there is more diversity in PFS 230, but this diversity is not in all the regions of this antigen. There are some regions that are conserved. So we want to understand which regions are actually impacting the interaction of these antigens with antibodies. So When looking at the entire genome of Plasmodium Falciparum in sub-Saharan Africa, this image shows us the prevalence and subgrouping of the various populations. Of particular interest is figure labeled C, where we see how the various populations are differentiated when looking at the entire genome. Here we see how the Ghanaian population, which is in light green, is differentiated from other populations like Nigeria, which is in red, and Gambia in green. So it will be interesting to see how these differentiations might be looking at these project's antigens, that is the PFS 230 and PFS 4845. So yeah, we see the 3D models from PDB for the two antigens that we'll be working with and how they interact with antibodies. We can see the antigens colored in yellow and the antibodies colored in pink. So it will be interesting to see how these interactions are disrupted by the introduction of the various mutations that we will identify in this project. Okay. So the impact of this project will be the effectiveness of transmission blocking malaria vaccines in Africa, and whether the development of cocktail malaria vaccine might be a better alternative to single epitope or single antigen vaccines. Thank you.
Thank you so much, Courage, for this wonderful presentation. I would now like to ask Dr. Harpreet to share a few words about this. Dr. Harpreet.
Hello, Courage. I think very nice presentation and a very good research question that you have. So I have one question. As in your second slide, you are mentioning that the previous antigens, the vaccine against the previous antigen, have a diverse nature. So in comparison to those antigens, so can you let me know exactly what is the size of these antigens and how these antigens actually become a very specific in comparison to the previous antigens against which vaccines are available?
Can you please repeat the question?
Okay, so as in your first slide, you were mentioning that that must be the second slide, I guess. So you were mentioning that there are previously the vaccines are available against different antigens that have very diverse nature.
Yeah.
Right. So
yes.
In comparison to those antigens, so how these two antigens produce either have a more specific nature, whether that decide by its size or exactly what is the characteristics that make them more specific.
I don't quite understand your question, but
okay, let me rephrase the question. The question is, sorry, Courage. The question is, you are mentioning that there is a specificity, so less genetic diversity for these antigens than the current vaccine, right? You mentioned the current vaccine is affected by genetic diversity. What about these antigens? What makes them more specific?
Okay. So these antigens are now being studied, but genetic variation has not been fully explored in them. So I cannot speak to that until after the study. I don't know whether I answered the question right.
I think what you're trying to say is that there could be some genetic diversity as well, but one of the slides he showed the data, and it shows that at least one of the antigens looks like has a much lower allele frequency than the other one. But that's one of the objectives for him to actually analyze whether the diversity is there and whether the diversity is specific to different regions and different countries, and what could that mean for the effectiveness of a vaccine that would be focused on transmission.
Yeah,
Okay.
yeah. Thank you.
Okay, so have any idea about size of in terms of amino acid or nucleotide? I mean, how long these antigens?
Okay, so yeah, I haven't. No, no. I haven't have an idea of how long the amino
I think
acids.
that is sometimes what is the size. I mean, uh, in some of the cases, I'm not saying that every case, if the size of a particular antigen or epitope is small, that become more specific. Maybe that is the point. Maybe you can consider it. Like if you look at their size in comparison to the previous antigens against which vaccines are available, right?
Okay.
Okay, I think very very good presentation. Okay.
Thank you so much, Courage, and thank you, Dr. Kaur and Ilya, for your feedback. Next up, we have Elipklin Emekashi, who is going to present on understanding interactions between malaria vaccine candidates and other proteins to inform more efficacious vaccine designs. Eli, you can go ahead and present. Thank you.
Hello, good afternoon, everyone. Can you all hear me?
Yes, we can hear you and also see your screen. Can you go into presentation mode, please?
Okay. I'll be presenting on a project proposal dubbed Understanding Interactions Between Malaria Vaccine Candidates and Other Proteins to Inform More Efficacious Vaccine Design. The work is being done by a team from the Noguchi Memorial Institute of Medical Research at the University of Ghana. One of the team members as follows: Dr. Benedicta Mensah and myself, Eliki Mamegashi, and our principal investigator is Dr. Anita Gansa. And this work is done in collaboration with the Pine Biotech team. The team is interested in the population genetics of malaria vaccine candidate antigens. So, like my colleague presented earlier, malaria is still of major public health importance, especially in sub-Saharan Africa, in which Ghana is part. And so far, there's been efforts to control malaria, which is the use of insecticide, mosquito sprays, and then drugs. But then these efforts are being hindered by issues of drug resistance and insecticide resistance. So therefore, we need, it's about time we get a vaccine, but we need a broadly effective vaccine against the various diverse strains of P. falciparum malaria strains we have in our population. Over the years, antigens expressed at the various stages of the P. falciparum life cycle has been targeted for vaccine development. In the P. falciparum life cycle, when a mosquito takes a blood meal, it injects what we call sporozoites into the bloodstream. And these sporozoites move from the bloodstream and infect the liver cells. And the process continues to infect red blood cells, and they develop into merozoites, gametocytes, and then the cycle continues again. However, these vaccine antigens have been grouped into three major categories. We have the transmission blocking vaccines, we have the anti-infection vaccines, and we have the blood stage vaccines. However, the most advanced currently is the LTS's AS01 vaccine, which has been designed based on Plasmodium Falciparum circumsporozoid protein. This protein is expressed on the surface of sporozoids, and it inhibits sporozoid motility and invasion of hepatocytes. Another antigen of interest in our study that we'll be looking at is PFAMA1, which is a transmembrane protein that is localized in the micronemes of merozoite and is released during invasion of red blood cells. And this characterizes the disease stage of P. falciparum. This study was conducted is in two parts. The first part was looking at genetic diversity of two major vaccine candidate antigens, which is the PFCSP and then the PFAMA1, which is apical membrane antigen 1. And we looked at these antigens in Ghana in 3 different ecological zones. And for malaria control purposes, Ghana has been divided into 3 major eco-epidemiological zones. We have the Northern Guinea savanna, we have the forest belt, and then the coastal savanna zones. However, transmission in the Guinea savanna is high, and then transmission is known to be high during the rainy seasons and very low during the dry seasons. Another measure to characterize prevalence of high malaria transmission is the number of infective bites from the mosquito per year, and it's called entomological inoculation rate. We gathered that in the northern savanna zones, we have 418 infective bites per year. In the forest zones, we have 866, and in the coastal zones, we have about 50 infective bites per year. So, like I said, these 2 antigens, which is the circumsporozoite and then the AMA1, are antigens of interest. We are looking at the circumsporozoite protein. We are looking at the diversity of this protein in Ghana because the vaccine currently has been designed based on a lab strain of Plasmodium Falciparum, not taking into consideration the multiple strains we have in our population. And the LTS has been designed based on part of the central repeat region, which is the image on the right, and then the polymorphic T cell epitope, and has been fused with an hepatitis B antigen to boost immunity. And then studies have shown that the CD4 epitopes are very polymorphic. But then those epitopes are also very important in mediating cell-mediated T cell immune responses. Also, for AMA1, we looking at the different domains and then how polymorphic these domains are in order to ascertain whether we can use this vaccine and this antigen to build a vaccine as well. So hypothetically, we say that malaria transmission plays a major role in diversity and potentially the efficacy. This diversity could impact how efficacious existing vaccines might be in a natural parasite population. So earlier studies conducted on PFCSP in our lab, we found out that especially at the C-terminal regions, we found out that we have a lot of haplotypes which are shown in different colors on the bar plot and their frequencies as well. Sorry. So we looked at 2 different ecological sites, as I mentioned earlier. We looked at Cape Coast, which is located in the coastal belt, and we looked at Navrongo, which is in the northern belt, to kind of ascertain whether eco-epidemiological factors affect the rate of diversity. And we saw that more diversity was seen in Navrongo than in Cape Coast. Also, we looked at specific epitope regions, like I said, the T cell epitopes, and we looked at what kind of amino acids are being, what kind of mutations are being presented in these epitopes, and how conserved are they or how frequent are they. We also did the same analysis for the PFAMA1, looking at 2 different epitopes, Begro and Cape Coast. So with all these epitopes, or with all these variations that we've seen, we want to go ahead and look at how it affects structural interactions and function. Specifically, we've already ascertained variations in PFCSP and PFAMA1 in Ghana. And what we want to do now is predict the impact of these variations in the form of amino acid changes in specific epitope regions, immune epitope regions, upon the protein structure, interactions, and functions. And for the PFCSP, we are looking at mutations at the T cell epitopes of the PFCSP and how they affect binding to T cells. For the PFAMA1, we are looking at structural interactions between this gene and then another protein called PFRON2. PFRON2 means Plasmodium Falciparum rotary protein 2. These 2 proteins form moving junctions at the external membranes of merozoites to be able to invade the host cell's erythrocyte. So we want to see if diversity of AMA1 can affect binding of PFRON2 and how this binding will affect entry into host cell host erythrocyte. Some studies have been conducted, and then we know the structure of PFAMA1. And furthermore, we want to ascertain if the binding poses some kind of structural conformational changes between the wild type, which in our case, we are looking at the pre-F3D7 as our reference strain, and then the various diverse strains we found in our population. When we have hydrophobic conformation, or are we going to see the same hydrophobic conformations when we bind RON2 that are variant strains in our population to AMA1? I want to also do that for PFCSP and its interactions with T cells as well. So the first part of our objectives is almost done. We have aligned amino acid sequences, and we have done some population genetic analysis. We've done some genetic diversity indices where we looked at nucleotide diversity, haplotype formations, we look at haplotype diversities, we look at spatial variations between our 2 or 3 sites in the Ghanaian population, and we also did some PCA analysis. We also looked at the patterns of amino acid sequence substitution, and we want to employ the same methods to look at other African populations and also populations from Thailand. So this objective outline will be used in other African populations as well. Zoning on to the second objective, which is prediction of impact of amino acid change in immune epitopes upon protein structure. We will first go ahead to predict and model 3-dimensional structures of our variants, and then also predict how they bind, especially let's say the PFAMA and how it binds to PFRON2, and also the PFCSP and how it binds to T cells. And we'll also see if the predictions or the potential impact of the substitutions on the structure or function. So in Ghana, we have 220 samples. Earlier on, we looked at epidemiological differences. So in Navrongo, we had 128 samples. In Cape Coast, we had 92. And we want to employ Malawi and Kenya for PFCSP because these 2 countries are also undergoing phase 4 RTSS clinical trials. So we want to see if we are going to get the same results in these 2 countries. And for PFAMA1, our earlier studies in Ghana incorporated 331 samples, and we had 150 for Begro and 181 for Cape Coast. We also want to look do the same analysis in Mali, Kenya, and Thailand. Our data type is FASTA sequences, and this data was generated in our lab at Noguchi. overall, the implication of this study is that it will add on new information regarding the antigenetic diversity of these two major antigens that we are talking about. And this will help us in what kind of haplotypes to include in vaccine design, whether we need a chimeric vaccine that will be including the most popular and the most frequent antigens. And also, it will help us know if antibodies or T-cell mediated immunity will affect these variations the same way or not. Thank you very much.
Thank you so much, Eli. Dr. Harpreet, please go ahead and share your thoughts on her presentation.
Hello, Ellie. I think it's a nice presentation, and you have already addressed the population that you are considering. I think it is very good project, and you can proceed for this. Yeah.
Okay, thank you.
Okay, thank you so much, Dr. Harpreet and Eli. So the next presentation is by Rita Boateng, who will be presenting on understanding mechanisms underlying host immune responses to severe SARS-CoV-2 infections in malaria endemic regions. Over to you, Rita.
Hello, good evening, everybody. My name is Rita, and I'll be presenting on understanding mechanisms underlying host immune responses to severe SARS-CoV-2 infections in malaria endemic region. And this project is a team project. So for the team, I'll be working with Francis, who is currently has finished his MSc in human genetics. And I'm also currently a PhD student working on structure analysis and computational biology. And for our team, we will be supervised by our PI, which is Dr. Anita. And also from the PINE group.
Hi, Rita. Can you make your screen in a presentation mode? Thank you.
Please, can you see it?
We still see your slides, all of them.
Okay, sorry.
Yeah, as Courage is writing, switch your display.
Okay, I've done that. Can you please see?
No, we just see all of your slides. I think you can continue if you are not able to do this. It's okay. All right, please continue.
Okay, sorry for that. Okay, so for my team, I'll be working with this group of people to make the project successful. So as we already know, malaria over the years is characterized by high incidence of the disease, basically or mostly in Africa.
You're still on your first slide.
Rita, if you are using two displays, then you have to show the one on which you are projecting your slides.
I'm not using two displays.
Oh.
Please, is there any difference?
We can see your slides. Okay, Francis says that he can share his screen. Yes, Francis, you can please go ahead.
Yeah.
I will just make you co-host. Give me one moment.
So I think when you share, you should share your screen itself, not the slide, not the PowerPoint presentation, so that if you project, we'll see the screen itself. Because if you share only the PowerPoint, sometimes it might show the non-projected slide.
Yeah, but I think at this point, maybe just, oh, okay, great. There we go. Excellent.
Can you see?
Yes.
Yes.
Okay.
Yeah, you can. Thank you so much, Karin.
Okay, thank you. My name is Rita Ifrieboati, and I'll be presenting on my team, with my team, which is on the topic understanding mechanisms underlying host immune responses to severe SARS-CoV-2 infections in the malaria endemic region. So this project is done in a team, and my team I'll be doing this project with Francis, and then also be supervised by Dr. Anita, and also the Pine Group. So as already said, malaria over the years is characterized by high incidence of the disease in Africa. However, with the emergence of COVID-19 cases, which is caused by the SARS-CoV-2, we could see a different trend. So the spread of the disease is, people are said, is considered less than like what we expected. Okay, so what we see is that even though we know that malaria is endemic in Africa, and as we were expecting that the emergence of COVID-19 could have some severe effects in Africa, but then we are seeing otherwise. And then we see that the high proportion of these asymptomatic cases and in malaria endemic regions as compared to other parts of the world. And the major findings have postulated that these major differences with SARS-CoV-2 infections across malaria endemic regions and also non-endemic regions is as a result of the severe stage of the SARS-CoV-2 infection. These severe stages of the SARS-CoV-2 infection is described by various distinct immune host responses, such as the inflammatory cytokinase release. And an example is the hyperinflammation conditions, and also cytokine storms, which actually provoke severe lung alterations that can lead to multi-organ failure. An example is the pre-existing malaria infections could actually be a factor to observe the level of severe infections, severe SARS-CoV-2 infections. An example to show is the low number of the severe cases in sub-Saharan Africa, which some research have postulated is as the reason why there is variable distribution genes such as the ACE1 and D, and also the ACE2 polymorphisms, and which have been reported to partly explain the observed clinical heterogeneity that we are seeing across endemic and non-endemic regions. So from this, it is important that as researchers, we should be able to investigate some of the underlying mechanism or factors that are contributing to this, and looking at the host immune responses to this phenomenon. So for our study, we hypothesized that this observed low reported case of severe SARS-CoV-2 infections that we see could be attributed to the high exposure of malaria infections in malaria endemic regions, and the adapted host immune responses to malaria. And for this study, our main aim is to investigate transcriptional signatures and molecular pathways involving in malaria and severe SARS-CoV-19 infections. So for the project, our first objective is to compare RNA transcriptional profiles of the various stages of malaria and also the SARS-CoV-2 infections, and we'll be looking at the adult population, and also to perform some functional annotations of differential gene expressions. So for the outline of our method is consists of 5 main steps. First, we select the data, then as defined the disease phases of interest that I've already said. And the second, we will clean the data and put the required formats for downstream analysis. And for this method, we will make good use of the Pine pipeline to perform some of this analysis. And so thirdly, we will perform RNA-seq analysis by leveraging the pipeline that's developed. And then fourthly, we would perform element-based analysis, which will include factor analysis and also regression analysis. And also, we will also perform some PCA and clustering and other statistical analysis. Then we would perform functional annotation analysis, leveraging the pipeline and also some important annotation databases available. So for our data set for this project, we have a data set of transcriptome data set for COVID-19, severe COVID-19 infections, which is 1. And this data set consists of expression profiles from high throughput sequencing. And our second data, which will be a comparative one for the SARS-CoV infection, is the data for malaria. And for this, we select the acute, the convalescent, and also the peak of the infections. So just to show you what actually is in this data. So for the acute malaria section, we have a total of 25 samples. And for there, we have the acute infection of the malaria, which we have 10. And then the convalescent stage, which we have 10. And also the peak infection stage, which we have 5. And also for COVID-19, we have the remission stage, which is the stage that there is a decrease in or a disappearance of the signs and symptoms of the COVID, although COVID might be present. So the factor of asymptomatic is also present. Then also we have a place where there is no remission. And for this, we have a total of 125 samples with 116 for remission and 9 for no remission. So for our study implication, we would like to see at the end of the project to elucidate some of the underlying mechanisms of severe SARS-CoV-2 immune responses in malaria endemic regions. And also from the analysis, we also wish to look at some significantly expressed genes which could be very good targets for COVID-19 treatment, being it vaccine development or novel drug targets. Thank you very much, and we are ready for any question if there is.
Thank you so much, Rita. Ilya or Dr. Harpreet, if you would want to share something, thank you.
I mean, I just want to ask, this sounds like a very interesting study, but have you thought of a control? So for example, Ghana or Africa is one of those regions with malaria, but also you have some Asian countries, or you have maybe South American countries that have a lot of malaria. Is it the same there? Have you seen the same trend of infections there?
Okay, thank you for the question. I think so far we've not thought about that line, but I think it's a very good question, and we will consider that for our next presentation, looking at the control.
Great, thank you.
Yeah.
So thank you, Rita, for presenting again, and to all of the participants in our infectious disease program. Thank you again for sharing your presentations and your projects with the group here. I wanted to focus and highlight on some very interesting topic regarding Africa. So we have been organizing Omics Logic training program in Africa since 2020. And now we have a dedicated team that serves hundreds of students across various countries in Africa. So as you see in my screen, these are some of the data that we have from the different programs we did in Africa. So we have had over different number of participants in all of these programs. So these are some of the programs that we have done with the participants there, and numbers who have joined these sessions. Here you see the countries in Africa from where we have the most number of our participants joining us from. So which is also interesting to watch, as all of these participants continue to collaborate with each other, they continue to learn together. And here is an interesting insight that we learned from our participants' background. So this pie chart shows you the different numbers of undergraduate, graduates, postgraduates, PhDs, as well as industry experts that we have among all the participants who take part in this Omics Logic programs. And also how many of them are saying whether they would be recommending the program to others. So we have had about 98% of these participants who say that yes, they would definitely want to recommend the Omics Logic training programs to their friends or their peers. And I also wanted to take this moment to introduce you to the Omics Logic Nigeria team. So this team is being led by Adekunle Adiloboye. He's the Omics Logic Nigeria program lead, and also faculty at Lead City University in Nigeria. Along with him, we have Aditayo Borisade. He is also a faculty at Bayero University and the Omics Logic Community Manager for Africa. So in the chat box, I would also be posting the email ID for Aditayo and the Africa team, which is africa@pine.bio. If you all want to reach out to them for any kind of questions relating to the Omics Logic training programs in Nigeria and Africa. And we also have Sani Hamza, who is a medical laboratory scientist. And together they have been running and conducting these Omics Logic programs in Africa. They are completely trained, and they also organize their own sessions. So please feel free to reach out to them regarding any kind of basic or specialized training that you are interested in. And now I would like to invite some of our participants who are part of these Omics Logic training in Africa to talk more about their experience in this Omics Logic program. So firstly, I would like to invite Oshuninka Damilola to share the presentation with us. Over to you, Damilola.
Good evening. Let me share my screen. Thank you, Pipsa. Okay. Please, can you share my screen?
Yes, we can see your screen now. Yes.
Okay. So, good evening, everyone. I'm Damilola Oshuninka, a graduate of physiology from the University of Lagos, Nigeria. I'm here to present a review about my journey thus far with Bioinformatics Omics Logic program Africa, a program which I actually first started about 2 months ago. So firstly, I would like to give a brief background about myself and what prompted me to join the program. So like I said, my name is Oshuninka Damilola, and I graduated from the College of Medicine, University of Lagos, Nigeria in 2019, where I majored in physiology, and currently I'm an Omics Logic training participant, and my core interest lies in drug discovery. So right from junior high, I've always loved calculations, but a higher curiosity drove my interest to major in physiology in college. See, I knew next to nothing about the human body, and I was determined to satisfy my curiosity. Believe me, I did. Furthermore, in physiology, I understood the mechanisms behind several pathologies, and my next goal after completing my bachelor's was and is to understand the steps that have been taken or are currently undertaken to cure these diseases. So perusing the internet on a certain day, I discovered that bioinformatics was just the perfect discipline that inculcates several disciplines, amongst which are my favorites, maths and biology, to answer questions about the mechanisms that drive population diversities, especially in terms of resistance to medication, as well as the genetic or phenotypic characteristics that actually cause these variations. Progressing with the course, my scientific goals are somewhat modified. That is, to drive prescriptive medications from a one-size-fits-all to personalized medicine based on genetic profile and transcriptomics expressions. Why? Because the robustness of our body's interaction networks leaves room for the formation of alternative network roots with the same disease expressions in different individuals. Take, for example, the triple negative and the luminal basal breast cancer. Also, I refine this goal because so much resources go into developing new drugs every year, and more than 20% fail at clinical trials. So I figure there is much more need for targeted approach towards drug development. Overall, Pine Biotech's training program has taken me several steps closer to my goals, and these are some of the notable takeaways from the lessons and sessions. I've learned about the basics of genomics, transcriptomics, the crucial role the microbiome plays in regulating our body functions and in determining the efficacy of medications we ingest. I also learned to use R and Python programming tools to read and visualize data, that is the sequenced reads, in the coding arm of the program. Notably, I understood the role of bioinformatics in biomarker analysis for effective diagnosis. I understood the classification of diseases into subgroups for personalized therapy using machine learning tools like principal component analysis. Moreover, I understood that allelic variants and mutation play a major role in population diversity and actually determine the drug efficacy in individuals. For instance, where you have a pharmaceutical company produce a drug and at clinical trials, some subjects don't respond to the treatment because of a nonsense mutation that prevents the full formation of cytochrome enzymes, and so prodrugs can be converted to active drugs. Furthermore, using the variant calling pipeline to detect single nucleotide polymorphisms, a picture of which you can see by the right side of the slide, I was able to determine if and which variants actually cause pathological defects in mutated cells. So these are some of the training paths we took and the pipelines I ran. Orion RNA sequence pipeline with prearranged algorithms such as Boltype-2G to map on reference genome. I calculated the abundance of reads using RSEM. I used quantum normalization to remove zero expression levels and to convert reads to logarithmic scale for further analysis using more machine learning algorithms like PCA. Going forward, these are the areas I intend to pursue, such as identifying biomarkers based on classification of gene expressions within a disease type and running simulations of molecular docking techniques and exploring how prototype drugs interact with proteins to block signaling pathways. Some of the scientific questions I'm interested in answering using bioinformatics skill set are: what are the mechanisms or characteristics that drive population diversity, especially in terms of our body systems? Does our daily interaction have effect on our genetic expression over time? How can we use the study of rare genetic afflictions to gain pioneer insights into prevalent diseases? So the first question that I actually had to answer was, during the sessions, I've been able to partially get answers to this when Dr. Adetayo talked about SMPs, and even from the metagenomics course I took on the T-Bio platform, I was able to see that even the human microbiome actually drives population diversity. The T-Bio courses are so well structured that participants can run demo pipelines solely on the steps listed. But of course, during the program, I had awesome mentors who took their time, even at odd hours, to explain difficult concepts. Mentors like Dr. Adetayo, Mr. Dikunle, Mr. Sani, amongst others. I mean, you'd be hard pressed to find support like that in other bioinformatics programs. I must say that this program has laid a really solid foundation for me to run bioinformatics related research projects confidently. And I look forward to continuing with the team to reach the next milestones on the program. Many thanks to my co-participants who made it fun, and thanks also to the coordinators, both on the national level and the international level. Thank you very much for listening to my presentation.
Thank you so much, Damilola, for the wonderful presentation. We wish you all the best. So for the next participant is Zainab, who is also going to be sharing about the experience of the Omics Logic program. So Zainab, over to you.
Hello, good evening, everyone. I'll go and I'll bless you. Please, can you share my slide? Okay. Hello, good evening, everyone. And today I'm going to be presenting about my steps to bioinformatics through Omics Logic training in Nigeria, Pine Biotech. My name is Zainab Absalam, and my name is Zainab Absalam, and I'm an undergraduate student of the College of Medicine, University of Lagos, currently studying pharmacology, and also a participant of the Basic 1 Omics Logic training. I'm passionate about computational biology and drug development. My scientific goal for participating in this program is to be able to incorporate data science and machine learning into science and healthcare, and my research brought me to a field called bioinformatics and computational biology. By participating in this program, I'm taking a step towards my career goal, which is to become a drug developer, and this program has totally gotten me on ground and got me started with bioinformatics. Notable learnings from the session: the training is packed with a lot of good courses like Introduction to Bioinformatics, the Transcriptomics, the Genomics. Basically, I've thought bioinformatics is basically genomics, but I'm starting the program to make up bioinformatics, other fields presented in it. Also, the code, the Python and the R programming language is user-friendly and can be easily navigated by a participant. Also, I was able to understand how it's being used in bioinformatics. I also learned the different formats into which biological data can be presented. That's the SAM files and the BAM files, and also the different repository where you can get your data if you have your personal project you want to carry out. My area of interest going forward, I have interest in computer-aided drug discovery, drug development, and computational biology. I'm presently working on mathematics in order to see whether an activity that can be incorporated into the discovery of HIV or COVID-19. And questions I would love to solve using bioinformatics is the incorporation of an African genome into the drug development. I mostly found out through research that Africans are not being inclusive. That's why in the recent time, the COVID-19 vaccine brought to South Africa, there was a complaint about side effects and all. And I'm going to advise anyone who hasn't registered for this program to please do so. It's very eye-opening and it's a very good program to get you started. You really don't have to be in the science field. You can be in any field, and the program is like, bioinformatics is very diverse and can be incorporated into any field that you like. Thank you very much all for listening.
Thank you so much, Zena, for sharing this. And next we have Dr. Nancy Unzulu share her feedback with us. Over to you, Dr. Nancy.
Hello, everyone. Can you see my screen?
Yes, we can see your screen. Thank you.
Okay. My name is Ifeoma Nancy Nzelu. I'm a veterinarian by profession, and I lecture with the Federal University of Agriculture, Makurdi in Nigeria. My specialty is actually in veterinary public health and preventive medicine. I was one of the participants during the recently concluded basic Omics Logic 1 training. My research up until this point, it's on zoonotic parasite, trying to understand the epidemiology. And recently I delved into genomics, characterizing the parasite that I worked on. That's on Toxoplasma gondii. It's been a long passion for me since my DVM days up to this moment, working on apicomplex and parasites. My research interest is on molecular epidemiology of infectious zoonotic diseases and in food safety. Now, how did I get into Omics Logic? I got this information from a Telegram group I belong to, the Nigerian Bioinformatics and Genomics Telegram group. Because of the play I had for bioinformatics, it prompted me to go in further. I actually wanted to do bioinformatics at the master's level, but I had no mentor at that point on what it was all about and how to go about it. In fact, a senior colleague I asked told me that you get to just sit down on a chair and you keep computing and typing and all that. So because of that, I didn't pursue, I didn't go further, but this opportunity came, and because of my flair for computing, statistics, mathematics, I went in for it. So far, I've been able to do some of the courses. This is my progress level for now. Though I have lots of work, I try to create time to do this. So I've been able to do some of the courses, Introduction to Bioinformatics, Genomics, Introduction to Genomics, Metagenomics, and also very interestingly that I love so much is the coding section. I've been trying to learn how to program using R and Python. And so this platform gives you the basic on how to use R in solving data or information. So it was very interesting and very simplified for me. So my scientific goals for participating in this program was, one, to learn about bioinformatics. Remember, I had a flair for it. Then to understand omics data and how to interpret it when I see it in peer-reviewed journals. You know, sometimes when you look at this paper, you see it's so technical that you can't even understand what's talking about. But now, after going through the first program, I could sit down, digest the paper, and follow it to its logical conclusion to a certain extent because of the basics I gained, you know, getting involved in the program. Again, I also wanted to apply bioinformatics to my area of specialization, which is veterinary public health. You know, marry it into epidemiology of zoonotic infectious diseases, and then identify a problem within this context and solve a research problem using omics data and its analysis too. Now, being involved in this program where I was exposed to a real-time, real-life, cloud-based server, the T-Bioinfo platform, where we ran pipelines analyzing omics data. We worked on variant calling. You know, we had to go through the various process. We had the pre-process, maps using Bowtie 2, and then did the differential variant calling using Strecker, then ended the pipeline, and then we visualized our results. We visualized the result on IGV. So it was actually an eye-opener. I could, you know, understand the process and make sense, make meaning out of it. Again, I was also exposed to loading and visualizing of data in R and Python, and understood the basic concepts of the omics, bioinformatics, genomics, metagenomics, epigenomics, transcriptomics, and then analysis of next generation sequence data. My area of interest going forward, I want to apply bioinformatics to epidemiology of infectious zoonotic diseases. How do I use this knowledge, you know, in understanding the epidemiology of these diseases? Is there anything at the gene level that actually predisposes a particular group of animals to a particular disease? Even among the disease group, across various age groups, sex group, is there anything about at the gene level that makes them being predisposed? This is what I want to look into in the future. I would want to also answer relationship between genotypes of parasites of interest and pathogenicity. For example, Toxoplasma gondii, which I worked on during my PhD, studies have shown that particular genotypes of the parasites are more pathogenic than the other. And so I want to look at this relationship, and then in the Nigerian context, in the Nigerian population, and also check evolutionary relationship between parasites within Nigeria and Africa as a whole, and that's obtained elsewhere in the world. I also want to explore the host pathogen relationship. It's going to be a new area for me because I've not actually done so much about this, but I'm open to it because going through this program has actually opened my eyes, and listening to the wonderful projects here, I mean, it's really an eye-opening opportunity. So the unique advantage with this program and why I recommend it is that first, as someone who do not have any basic knowledge at all of bioinformatics, you are able to understand the basic concepts of molecular biology and bioinformatics. And also the program and the platform also gave great mentorship and support to the participants. For example, I had a situation where a data was with me till past 11, trying to take me through, that's past 11 in the night, trying to take me through how to visualize the genome using the IGV browser. I mean, it was interesting and surprising that I could get him on at that time. And I had my baby by my side, but we went through it and I got it done. So the mentorship was superb, the support was wonderful. And then they have well-designed courses with great content. Yes, I give it to this program. They have great content. And then you are able to assess the knowledge you have gained so far by the quizzes, the design of the quizzes. In fact, in some situation, I was sweating answering some of those questions, and my blood pressure was high because I wanted to, get the 80% pass mark. So it was quite interesting, and more importantly, there were hands-on practice with the T-Bio platform, with the T-Bio server. If you ask me, The next level for this program should be a degree awarding program, because having gone through these stages, I think the leaders and administrators of this wonderful program should be thinking towards this direction of participants actually getting a degree after going through this training. And I want to say thank you to the Omics Logic Nigeria team and to Pine Biotech for this wonderful opportunity. And I intend to move further with the course. Thank you very much for having me.
Thank you so much, Dr. Nzulu, for sharing such a wonderful feedback with us and your experience. It's wonderful to hear from you. We also know that you have been very actively participating in all the sessions and understandable with all the work that you have. So thank you again for participating. And now the session is open for all questions. I again want to thank all of our presenters today who took the time to prepare this presentation and have been working on these several projects, several different programs. So please feel free to ask any questions that you may have. And you can continue to post those questions right here in the chat box. And also, if you all want to be part of similar programs like this, as I'm not hearing any questions, I would be introducing you to some of the upcoming programs that we are having. And also, as we are going forward, let me also introduce you to our groups in LinkedIn and Facebook. So these are the groups that we have for all of our participants, or even those who are not part of our training program but would love to be members of our community. We have these two groups in Facebook and LinkedIn that you are welcome to join. And here are the upcoming programs. So for the first program, we have a training which is starting this Friday on OmicsLogic Genomics. So this program is again part of the 6-month training program that I had mentioned in collaboration with Noguchi Memorial Research Institute at the University of Ghana. So as I mentioned earlier, that we would be having for phase 2 genomics and then transcriptomics. So the first part is the genomics program, which is starting this Friday, which is March 12th, and the time is 3 p.m. GMT. You can convert that to your own local time zone. And this would be a 1-month training program, which is open to every participant who is interested in learning about genomic data analysis. So please feel free to ask us regarding the training program, and I will also be sharing the link for this program very shortly. And the next program that we have, which is starting on March 15th, is Bioinformatics for Precision Oncology. So this is again a 1-month program on precision oncology or for cancer research. So this program will start on March 15th, which is next week. And here again, anybody with an interest in cancer research can join and take part in this training program. So let me also share with you some of the links to this different program. Okay, so let us first start with OmicsLogic Genomics. And thank you, Ilya, for sharing the links again. So this is the link to pre-register for the program. So once you're pre-registered, you will receive details regarding the program. We do not have a lot of time. As you see, we are starting on Friday. And regarding this, so this is the Precision Oncology program. So please feel free to review these programs. And if you have any questions, please reach out to us. For participants who would require scholarships, please, you can also write to us regarding this at marketing@pine.bio regarding any of the programs that you are attending and the scholarships that you would be requesting. The only thing that we want to mention here for anybody who is applying for a scholarship is that we would need to meet with you and one of your references if we are to give you the scholarship for the programs. And during the meeting, we will walk you through the different steps, processes, and also introduce your reference to this opportunity. And based upon that meeting, whether you receive a scholarship or not is decided. So please make sure that you're mentioning the the references as well. And it's very imperative that we set up the meeting with both of you. So please make sure that you're following this, and you can reach out to us either at marketing@pine.bio. And if you are in Africa and from any of the African countries, please feel free to reach out to africa@pine.bio. Our Africa team works very closely with us, and they would be able to help you and connect you to the right resources, and you can also chat with them. So thank you, Aditayo, for sharing this. And yes, for Precision Oncology, we also have a webinar today at 5 p.m. in US CST time. So if you're in that time zone, or if you feel that that is quite a good time for you to join, you are welcome to request us to fill up, or please fill up this form here, and we will be sending you the Zoom link for that session. So thank you again, everyone, for attending today's session. I do hope that you enjoyed this session, and I look forward to having all of you with us again. Have a wonderful evening.